Okay, hello everyone. Greetings to everyone who joined us today. I'm Christelle Rabil, and on behalf of the Crop Trust, thank you for joining us for our How to Use Genesys webinar. Today's webinar will be given by Mattia Obreza, head of the Genebank Information Systems at the Crop Trust. Mattia has extensive experience within the field, having started working with the Genebank data in 2007 at the International Institute of Tropical Agriculture, IITA, in Nigeria. So today, Mattia will demonstrate how to use Genesys with a focus on passport data. And after the presentation, there will be around 20 minutes to answer your questions in the Q&A box. And with that, I hand it over to you, Mattia.
My name is Mattia Obreza. I work at the Global Crop Diversity Trust on Genebank Information Systems, and will really try to race through this presentation to leave as much time as possible for your questions and answers at the end of this presentation. So if anything pops to mind, please use the Q&A box, and we'll try to get to all the questions at the end. Right, so with that, this session sort of will give you a quick introduction on what Genesys is, the sources of data. We'll have a look at the Genesys website. We'll talk about the accession passport data in Genesys and the MCPD, so the multi-crop passport descriptors, in very brief. And the bulk of this presentation will be focused on browsing and searching Genesys with practical examples, followed by your Q&A. So Genesys is an online platform where genebanks publish information about the crops maintained in their collections. So Genesys does not host information on in situ germplasm, breeding materials, or released varieties unless they are formally part of ex situ genebank collections. It is our vision that with Genesys, detailed accession-level information is easily and freely accessible to users of germplasm. And to that extent, we support genebanks in documenting and preparing existing data for publication in Genesys. We work with established networks and individual genebanks, and we also work very closely with international and regional genebanks across the world. Right now, without further ado, let's look at Genesys directly, so we can spend more time on exploring Genesys and its functionality, and less time on the intro. So I'll stick to the slide for a second. Genesys is accessible at the URL you see down here. You can scan the QR code on your mobile phone to access it on the mobiles. Anyway, the easiest way to find Genesys online is to just Google for Genesys PGR, and you're going to hit the Genesys website. Now, to explain some of the layout of Genesys website, you have the top navigation bar that's always visible. That basically provides you the main navigation options to get back to the home page, to the entry page of this page. Explore different sections of Genesys, some information about Genesys itself, the documentation, the selected list of accessions that you've created yourself, and a login button, hopefully, or your user profile. The main search box here is a full text search option, followed by some numbers of some statistics on the numbers of accession records available in Genesys. This is the active accessions currently in Genesys, different subsets. So that means core collections, mini cores, reference sets, and trade data sets. Then you have recent posts that are updates prepared by us in collaboration with the partners, and click on them, you can read the full post. After that, we have a selection, actually a random selection of a few crops in Genesys, and a link to browse all crops that we maintain, and so on and so on. Right. So before we look at the, how to make any sense of the 4 million accession records in Genesys, I just want to take you through the data that's available for one single accession in Genesys, and I'll be looking at an accession I picked happens to be an accession from the Erie Genebank. I just hope I can navigate my way over to Genesys. Link. I'm just going to go directly to that accession and try to explain what this thing is all about. So this is basically the accession passport page of one single accession. Every accession in Genesys gets its own unique URL. So if you link to this, you will always get to the accession details, where you can find the information as published as the Genebank, based on multi-crop passport descriptors that we'll cover later. So you get the information about the accession numbers, which Genebank is holding the material, the acquisition date, and so forth. And Genebank can, in Genesys, attach a number of images to one accession. That those images will be displayed here. So by clicking on the image, you get the full details, you get the full original picture displayed. In this case, we have one image, but it's possible to have as many as Genebanks decide to upload. So the important part to highlight here is the taxonomic information of this accession. This is the bits as provided by the Genebank. So this follows MCPD standard. And we do try our best to link up the names, the taxon provided by the Genebank, and map it to a taxonomic backbone provided by the Green Taxonomy database maintained by USDA and PGS. The section of accession names, this is where you would find all the different identifiers and names for this specific accession that the Genebank knows about and makes accessible to Genesys as well. Now, going further down, this accession apparently was originally collected from its in situ environment, apparently from Turkey. And as soon as Genesys has coordinate data, it will place a marker on the map of the world to sort of highlight where the accession comes from. Now, there's all fun things we do with coordinate data, because once we have that, we can link it up with climatic data. So we can show the climate at that origin, so at that locality, and display the various parameters. And these are all sourced from the WorldClim data sets. So with that, the last section on this page, I'm going to skip some buttons. The last section of this page is the passport data completeness index, and this is a measure of how complete passport data of this one accession is. And I'll return to this as soon as I show you some MCPD slides. Right. So passport data in Genesys follows the multi-crop passport descriptors, or MCPD for short. This list of descriptors was originally developed by IBRI, Biodiversity International, and FAO, and it's still occasionally updated. So if all accessions in a Genebank collection are documented using MCPD, the resulting spreadsheet looks something like this. So each descriptor in MCPD corresponds to a column in Excel. So it's very important to know and understand what is meant by each header, by each descriptor, to make sense of the data and to actually be able to make proper use of this data. And you can find the descriptor definitions also in the Excel file if you download it from Genesys, or you refer back to the MCPD version 2.1 that we linked to earlier. So also this format is also used by Genebanks to provide passport data to Genesys. MCPD can be clustered into seven groups. So we have the identity at the Genebank, taxonomic information about the donor institute of that material, so who provided it to you or to the Genebank, information about where the accession was originally collected from its in situ conditions, breeding data in case that is a breeding line, or if the accession is developed or was bred, and the geographic data, which is very useful because from this we can link up to various different other data types, including soil, climate, and other GIS data, just based on the correct latitude and longitudes of accessions. There are various status flags in MCPD as well, and I'll just highlight that there's one we use in Genesys, and that is whether the accession is an active accession or a historical accession. So it's if it still exists in the Genebank or if it was thrown out. And I think we'll explain some of that historical stuff in one of the use cases later on. Right, so this is basically the MCPD descriptors. And with that, I can return back to my IRGC accession. And as you see here, this PDCI score, all these little labels here correspond to MCPD descriptors. And depending on the SAMstat, on the biological status of the accession, various different descriptors contribute to this score. So if the accession, for example, was a breeding line, then you wouldn't expect to see collecting site latitude and longitude, and they would not contribute to the PDCI score of such an accession. But for things that are traditional land races or traditional cultivars, you really want to know where they were collected from, right? So in that case, collecting data and the site contributes significantly to the PDCI score. Right, so I want to scroll up here and show you one of the tools available to you in Genesys. Just because this accession is currently flagged as unavailable for distribution for various reasons, and I need to log in first before I continue with that. So let me just log in. So for users that are logged into Genesys, these two buttons show up, right? So I'm going to click the similar, so this button that says show similar accessions. And what this does, it will execute a search across the 4 million other accessions in Genesys to find potentially matching records in a different gene bank. So on the left side, we have our initial IRGC 3209. And the best match that was found by Genesys with this algorithm is an accession in USDA that has this PI 16498 something, right? And it displays the two accessions side by side so that you can actually compare the data and assess the similarity of the two by yourself, right? So this is really not something an algorithm could do. It's really up to the curator. It's up to you to decide if the two are actually the same accession, right? Now, in this case, because we know it came from USDA, and we have a PI number from Erie, this PI number actually does correspond one-on-one to the accession in USDA. So these two accessions are very similar, if not originally the same. It's just they're just maintained in different gene banks. And as you scroll down, you can see the passport data available in one collection and passport data available in the other collection. There are multiple other potential candidates for similarity, so you can navigate between those by using these buttons on here. So that was on similarity. Another button that is available for a user when looking at one accession is show changes, and that just displays when changes were made to individual fields of this accession through time. And we do clean up the audit logs every now and then so as to not, overflow, because every change does get recorded, and then we want to keep the database small, and it's also that the change is also not that relevant after a few years. Right, so going forward, we'll actually go to Genesys, and for that, let me just hit a reload here. So I want to look at lentils in Genesys as an example. So I'm going to go through crops and crop groups, and I'm going to be lazy and just search for lentil because I don't want to scroll. And I'm going to click that, and we get to a Genesys crop page for lentils, and the page provides some basic information about lentils, but more importantly, it shows what Genesys knows about lentils in its database, right? So you can access the passport data of lentil accessions through this browse accessions button, or by clicking the number here. So there's about almost 31,000 active accessions in Genesys of lentils. Further down, you'll see some generic images for lentils and some overview numbers of which gene banks maintain them, what species are there, and so forth. So I'm interested in the list of lentils, so accession data of lentils. So I'll hit browse. And with that, Genesys opens up this accession browser, and it says that it found about 31,000 accessions for lentil as a crop, and it is currently excluding historical data, right? So I could enable that, but I'm going to leave that out for now, right? So this list provides links directly to individual accessions. So if you click on IG4, you get the passport data of this one accession. The the functions we saw before are still available. You can try those out here as well. If the accession is georeferenced, you would see the map and so on, right? So that is very simple. Let's go back to the list. So here on top, you have the filters bar. So this shows you the currently applied filters. So it says we're looking at lentils, only active accessions. And the left side of the screen is what we call the filtering panel, where a number of filtering opportunities are displayed. So you can look at the holding instituted accession number, search by date, and so on and so forth. So looking just at the filtering panel and these 31,000 accessions, Genesys says there's about 14,000 accessions at ICARDA Genebank. So how do I filter out these accessions that are at ICARDA? So by clicking on that label for LBN 002, this would now show me, this would apply the filter and it would search lentils at ICARDA, right? So there's 14,000 some lentils at ICARDA, but I wanted just the opposite. I wanted to exclude ICARDA accession. So if I click this little plus thing, it changes to minus. And if I apply the filter again, it now says it's looking for lentils, active accessions, and it's excluding the records where the holding Genebank is ICARDA, right? So that is quite helpful. We got now the remaining 16,000 accessions, and we can look at what other options we have down here. We could basically look at the, let's say, the origin of material, for example, right? And select, add Turkey to the list, Syria, Iran, and hit apply. And with this, we are now looking at accessions of coming from Turkey, Iran, Syria, not at ICARDA, of crop lentil, and still excluding historical records, right? So I'm going to remove some filters. I'm going to click out Iran, Turkey, and Syria, focus on Turkey. And I'll apply my filters again, right? So there's 2,400 accessions, a bit more. And I can download the MCPD of this accession list from here. So if I hit this download MCPD button, I'm going to get that same Excel spreadsheet that we saw earlier, but only for the accessions that are listed here. So the Excel file is actually generated on the fly for every user for their specific filters, and it always includes the current data as is in Genesys. Right. So what else did we say? Okay, so let's have a look at the overview. The overview is basically like it explores these 2,400 accessions for the ones we filtered out. Let's drop Turkey. So everything not at ICARDA and lentils, and it tells you and how these accessions, how they are basically distributed or clustered. So we know they're all lentils because we're searching for lentils. But what we see here is the different species provided to Genesys. You can, look for those specific names. You can focus on traditional cultivars or land races. You can focus on things that are in different types of seed collections. And you can see here the different provenances. So you could say, okay, I really want only the accessions from Turkey. So if you click the number, it is going to add that filter directly to your filter pane, and we're back to our 24,000 accessions. Now with that, I want to look at the map of accessions. So this is still the same filter. So lentils from Turkey, not at ICARDA, and we get 258 matches, right? And these are the accessions you'll see here. By clicking any of the green dots, you can discover what's hiding under there. So if you open that, you get to the accession details page. And what I'm going to do now is actually, I'm going to show you something here. I can, sorry, let me just do that. I can save this map, and I'm going to call it lentil Turkey. Sorry, Turkey minus. Oops, that's not working. 1, and I'm going to give it this color, and I'll save. And now you'll see that there's a list here with this color I selected. I can toggle it, and this is my current selection corresponding to the filters I've had. Right? Let's disable this one. Okay, so if I now want to say, fine, we've done our filtering. Now let's only look at the card accessions, again, from Turkey. So I'm going to just flip this to plus. So that is going to give me lentils from Turkey, but now looking only at the card accessions. And if I enable the ones I had earlier, so without the Icarda, you get a little hybrid map with 2 different colors. You can change the opacity of one on the other to show how what is in Icarda, what isn't, and to compare those side by side. One quick thing here, since we're looking at the map features, you can download the data in KML format for these 500 accessions. So in this case, Icarda Turkey lentil. You can download KML, put that in Google Maps or other software and do your own GIS stuff. Or you go back to accessions list and download the MCPD of those accessions and do your own analysis, right? Right, that is on the maps. Let me see what's next. Right, so another thing I want to show you is how to look at the different, how Genesys handles taxonomic data. So with that, I'm gonna go to tomato. And what you'll have here is, okay, again, some description about tomatoes, the known buttons to browse accessions and to look at the accession passport data. And what you'll see here is that for tomato, we have, Solanum lycopersicum, but also Lycopersicum esculentum as the species names that fall under the tomatoes, right? So there's different species that contribute or that are grouped into this tomato crop, right? So let's go back to the tomato accessions and just browse all of them. Right. Okay, so here again, I can download passport data for these tomato accessions. I can look at the map, but since it's a lot of records, I first want to start with the overview. Right? So here, if we look at the composition of this data, the different holding institutes that have tomato, by clicking the blue other link, the list expands and loads more records so that you can actually look at the other ones. And here, what we want to do now is say, fine, we just want to focus on Solanum lycopersicum, and I'm going to click the number, same as before for Turkey. So if I click this number, I am effectively applying a filter for Solanum lycopersicum. I don't want tomato anymore, across all Genesys. And with that, I get to about 50,000 accessions. And one thing that you'll notice now as we scroll back towards the species list is that the species names of accessions as provided by Genebanks include Lycopersicum esculentum, even though I search for Solanum lycopersicum. And that is because the taxonomic backbone based on green taxonomy does help us, find synonyms in the search when searching the database. So if I untick include synonyms and apply the filters, the results are going to look a bit different, and no Solanum, none of the Lycopersicum esculentum is not going to show up anymore. So, like if I want the synonyms back, I need to enable synonym in the search, and they will show up again. So, if I focus only on the Lycopersicum esculentum, and I have to remove Solanum, what we're going to see now is under the species name provided to Genesys, we don't see the synonym of Solanum lycopersicum anymore. And that's because this matching actually just goes one way. So if you search for the currently accepted valid name as per green taxonomy, it will include the synonyms. But if you pick a synonym, it won't include its currently active name and expand the search. For various reasons, but we can touch upon that later. So that was on the taxonomic backbone, and it is pretty useful because it does address issues with different spellings of taxa by different gene banks, different synonyms, and allows users to still find the accessions they want by using the currently accepted taxa. Right. Okay. So we were focusing on accession passport data. So let me show you some more stuff. So on the Genesys entry page, there's this big full search box, and this executes a full text search across the Genesys database, and in this case, only focusing on accession records. So I'm going to search for, red dragon, just because that's a good example for, I like dragons. Right. So by searching for, red dragon, that same query is actually the one here on the full text search, and this executes really a search across where this phrase, these keywords could appear in the accession data. So you'll see that we do have a red dragon, but the first hit is a black dragon record, and if I really just want to see the red dragon accessions, I'd have to quote those, because that means search specifically for that phrase. And if I just search for dragon, I'm going to get a number of different accessions across different crops. So if I want to know what that looks like for those dragon accessions, let's say there's some wheat, barley, oats, and I just want to look at wheat. Do something here. So only want the wheat accessions. If I go back to the accession list, this is what I wanted to do. I can actually, add or remove. I can add accessions of interest, the dragon accessions of wheat to my list. I'm going to like this one, I like this one, and I will take this accession from Simit, this one, and some more, right? So with this, I've actually just flagged a few accessions for whatever purposes or means, and they get aggregated under this My List menu. So if I go to My List, this is where you can find then the records you've clicked, add to My List or not. So if I go back to that accession, this will add it or remove it to the list, right? So why is this thing useful is because you can make, a selection, and from here download the passport data again for the ones that are selected. And you can also initiate a request for material via Genesys. And by clicking that button, Genesys is going to inform you that of the selected 5 accessions, 3 can be requested through Genesys. And that's because only select gene banks opt in to allow for this facility through Genesys. So you know that the green ones are the ones you can request. So if you take the next step, you fill out the information, and Genesys will help you request for the material from the coding gene banks. Right. Okay. Now, so let's look at some use cases. I think we're doing pretty well on time, so that's great. Right. So one thing I wanted to show you is explain a bit about the historical records. So I'm going to look for only historical records in Genesys, right? Now there's a number, the menus are all the same, the data is all the same. It's just that the accessions are all flagged as historical, so they will all show up as red, highlight as with a highlight. And from here, if I go to the overview, just want to give you an example. I want to look at historical records from gene banks in the UK, or more specifically, I want to look at historical records at Q, and even more specifically, I want to look at historical records of GRASP accessions, right? So I've narrowed down my search, and if I now go to the accessions tab, there are the 15 accessions of GRASP at Millennium Seed Bank. And if I take this accession, for example, right, it is displayed as historical. So there's all these warnings all over the place. And this is where the similarity search tool is very useful, because if you do that here, you may find that the same accession actually does exist in a different gene bank. So in this case, this accession Aticarda does seem to share some identifiers with the historical record XQ. It does share the same elevation. It does share the same collecting date of sample. So it is kind of likely that that's the same material. So it is very important, and we encourage the gene banks to continue sharing data on the historical records, even if they have lost the material, because if there was research done on an accession and it does happen, you don't have it anymore for various reasons, there is still ways to get your hand, so that for people to find a corresponding accession in perhaps a different collection. So going back to exploring all accessions, let me show you some other stuff. So one of the use cases that we commonly have is how does one search for a list of accessions? So I have a list of accessions prepared here. I want to search, not by full text search in this case. I really want to focus on accession numbers. And if you copy paste the numbers from, actually, let's do it from here. Let's take these. So all I did here was select a few accessions, and so I guess copy these and drop them in accession number field. Genesys will convert them, into a selection of different accessions. And if I apply the filters, I should get back the accessions that I've selected here, right? So maybe even including historical, just in case, right? Or only active. So this is a simple way to find or actually add or search for accessions. Just keep adding to this list, apply the filters, and find the right stuff by resetting the field, and then download accessions, review them on the map. And these, sorry, we still have our fancy map enabled. So these accessions do actually correspond to something we downloaded earlier, right? Okay, so another useful use case here is if I wanted to find cassavas. Now let's say I want to find maize growing at high elevation, then I need to look for maize, and I need to look at maize only. So I'm going to pick maize. But high elevation, that means something above, let's say, 1800 meters, 1850. I have an example here. So I'm going to scroll down to the, the collecting data, and I'll say stuff collected at elevation of at least 1850 meters. By hitting enter, this filter is applied. We expect to then see a search for maize where elevation is above the value I keyed in, only active accessions. And we get the following matches. Now, the interesting thing is if we go to the map, what we can spot here is all these accessions do seem to come from the Himalayas. They come from the Andes. Juan Carlos could help me with geography of Mexico, but let's skip that part. Right, so this was a simple way to search for maize in high elevation. Let's do something else. Let's look at not maize in this case. Let's look at lentil. Lentils and wheat. This is a different example, also growing in high in highlands over 2,000 meters. Let's pick that out. So we do expect the wheat and lentil as crops, or wheat or lentil as crops, elevation over 2,000 meters, and on the map they will probably be geographically in those same areas, less in Andes, but much more in the Himalayas and Ethiopia. Right. One more example that I had was looking for cassavas from dry areas. So let's, actually go back to session list. I'm sorry. I'm going to clear my filters, and I am going to look for Maniot as a genus. So let's do. I'm just going to apply the filter first. And I am going to apply a filter based on the climatic parameters. And those climatic parameters are all aggregated at the bottom of this filter pane. So if you just scroll the mouse down to the end, you get climate at origin filters. And you would search for the annual precipitation, and something dry would have annual precipitation below, let's say, 800 millimeters. If we hit enter button, the filters applied now are annual precipitation less than what we specified. Accessions with generic name Maniot. Genesis found 800 accessions. Now, because we use a climate filter, that means this only considers accessions that come with coordinate data. I mean, because otherwise we cannot tally it up with climatic parameters. So if I look at the map, these 800 accessions, 800 some accessions, will have coordinates. They show up on the map, and you could basically scroll in and find the details of those accessions, or look at the overview and see the Maniots in what gene banks. And let's say you focus on CIAT or Embrapa, you can find those records there as well. Okay, some more use cases quickly. One of the things we do on our end often is to check what is most recent in Genesis, or actually, yeah. So if you want to see what the latest additions to Genesis were in the last, let's say, this month, this actually informs me mostly who's been active. We have a lot of new data from Embrapa, Ghana, World Veg, and I think this was IITA. Alternatively, we can only look at what was modified, let's say, in the last week. And in the last week, about 64,000, almost 65,000 accessions were updated. So again, World Veg and a few records from IITA, and the similar things are still available. This is really something that it helps me understand what has recently happened in Genesis. Now, usually what people want to do is explore just data on their own, look at the data of one particular gene bank. So in this case, this is the landing page of IITA. And here, there's a series of buttons, probably not all of them visible to just logged in users. A lot of these functions are available to IITA staff only, but the download MCPD button will be visible for the gene banks. And this is a download of passport data in Excel format for the IITA collection. Now, this is without limits to the number of records. So why that is important is when you have a gene bank that has over 100,000 accession records in Genesis, downloading those through this interface will not work, because we do have a limit of how many records you can download in one go. But if you do come from an institutional page and say, I want to access all records of IITA, you can get that stuff by downloading the passport data directly from the IITA page in Genesis. Right, so no limits on that. Okay, so I'm gonna try to wrap things up a little bit. So we did some use cases. Now, Genesis APIs allow for interaction with the data in Genesis, and all of the website that you see and you can interact with is built on top of these Genesis APIs. So basically everything the website does is accessible through the Genesis APIs. We have also BRAPI, so breeding API endpoints available, and there's a package for R, the statistical analysis package, that provides functions that wrap around the Genesis API to facilitate access to the data through when you are doing your analysis in R. The alternative always is download the passport data in Excel from Genesys and then do your analysis in the software of your choice. So just to wrap up, we're here to help. You can find us at helpdesk@genesispgr.org. And with that, we'll go to Q&A, and I'll try to do my best to answer your questions, if any. Right. Well, you want to look at the questions?
Yes, thank you, Mattia. There are actually 23 questions, and we'll try to cover all of them with the remaining time. The first question is, what are the evaluation criteria for the similarity of accessions?
Evaluation criteria for similarity of accessions. Right, so we do have to go back here. We actually do have a post about the similarity search. It was sometime last year. Oh my God. Searching for similar accessions. So you can find that. It's explained in this post. So under the home, all posts, just search for similarity and it's going to pop up somewhere down here. So that should take care of it.
Okay, thank you. Second question, using the similarities, can the Excel file be downloaded from the similarity search?
Right, no, so the similarity search at this moment is available only for a single accession. That is because the similarity search is a very expensive function. So just to calculate similarity of one accession across all the other accessions in Genesys, so the all 4 million other accessions, it does take a few seconds. So if you want to run that against, let's say, 1,000 accessions, you're talking about, quite a lot of time that Genesys machinery needs to do. But if you do have that need, we can organize for something and maybe generate an Excel file of those just for your case, but we do it not on the live Genesys servers, but on an offline copy.
Okay. Another question is, do you need to be logged into Genesys to download MCPD files?
Right, you do have to be logged in for same reasons as the similarity search. The reason being, downloading generating the Excel file is also much more resource intensive than generating CSV. CSV it comes with no formatting, but it's very difficult to assure that all the rows tally up, and especially with large data sets from many different gene banks, it's a high probability that the CSV will not be parsed correctly by Excel or whatever software then. So we do encourage people to download Excel, and for that you do need to log in.
All right. So there's another question. Can a user change the language to facilitate his or her search in Genesys?
Genesys today is available only in English. It used to be available in the different languages, but with the pace of development of new features in Genesys, translations to Spanish, French, Chinese, and so on, were lagging so far behind it, and they were not of quality that was acceptable. So we decided to actually switch and make this accessible only in English. Embedded Genesys, on the other hand, which is a different tool altogether and not part of this discussion, is a much smaller, uses a much smaller set of translations, and that is something that is much easier to translate into local languages.
All right. There's another question about the representation of NUS and emerging crops in Genesys. Are there any specific efforts to better represent these species?
Genesys makes available data that as the gene banks provide to us, right? So the more gene banks we get into Genesys, the more diverse the accessions in Genesys will be. So as we add gene banks, or as more gene banks join this, finding those the species of underutilized crops and other wild relatives of cultivated crops will become accessible. But this is really something that we cannot make happen. It really depends on the gene banks, what they maintain in their collections, and if or not they share the data in Genesys.
All right. We have another question since we're also on the map slide. Is it possible to integrate land use data to the accessions on the map?
If it's possible to integrate
Land use data or any other data sets to the accessions that are mapped.
right, so we do have a few layers built into Genesys. These layers are all generated from the world CLIM data. So if you take precipitation, you can actually enable an additional filter, which here doesn't really make a lot of sense because it's so you can see things. The trouble with additional GIS filters is that they are usually licensed. So if you want to generate crazier and fancier maps, you do have to get your hands on a proper data set and maybe generate the maps using your GIS package. So, and we know layers exist, the GIS data is out there. It is really a question of whether we can make use of it directly in Genesys and integrate it.
All right, so another question is, can we use the Genesys data of a particular crop for publishing status or policy papers? And if so, please elaborate the procedure on the part of the user for publishing the paper.
Right, so the use of Genesys data is governed by terms and conditions of use. This should explain, sorry, that was a bit quick. This should explain what you can and cannot do with the data. I think the main need here is attribution of the data source, which is the Genebanks that provide data to Genesys. Keep in mind that Genesys does not have all accession data available, but it does have quite a lot. So you'll usually have to mix it with other additional sources of data, maybe go to the national gene banks or another institutes in the country or region to get a complete set. Ideally, that would be available in Genesys at some point, but yeah, using data from Genesys is covered by the terms and conditions of use that you can find here. It's actually in the footer on everything.
Since we're on the use of data for publication, what about the historical accessions? Are they governed by the same rules? Can they be used for publication?
The records, the passport data of historical records, it's all governed by the same. The data itself, access through Genesys, is governed by the same terms and conditions of use. So when you download data from Genesys, just make sure you are looking at historical. I mean, that you pay attention to that flag, and if you're interested in what really is in the collections, make sure you filter out the historical records. Primarily, we keep historical data here so that it can be linked to alternatives in other collections. I would not use that to say, oh, so many accessions have disappeared. That would be quite a stretch, because accessions get uploaded and flagged for different intents and purposes.
All right, so another question is for the mapping. Is it possible to filter by altitude range? Example, between 2000 and 2500 meters.
Right, so right, so let's take wheat again or something. No, what did we take? Maize. Maize. Sorry. So let's look at maize quickly, and we'll put maize on the map. Now, since you are looking at maize in Genesys, the filters are still here. So if you did want to apply additional filters, all you need to do is say, fine, I want to use, an additional filter that deals with the location, and say elevation. What was it again? I don't know what the numbers were, but let's say at least
2000, 2500.
Okay, let's say 2000, 2500. Let's see what happens. Right, so there you go. And again, you can download the KML file for your GIS software, or take the accessions passport data from the accessions tab.
All right, thank you. So another question is, the annual precipitations are not in the passport data. So how does Genesys show this list? Right,
so looking at the map. So I mentioned earlier, when you have coordinates of where the accession was collected, then you can do a lot of fun things. And one of the things we do is actually take those coordinates and link it with the WorldClim data sets. In this case, for this locality, we are able to get the climatic parameters from the WorldClim dataset that correspond to this little dot. And since we're on this page, I'm going to show you something here. So for any georeferenced accession, we display the climatic parameters, but also there's a little button here that says, show me accessions that have similar climatic parameters. So let's click that here. And it has found 10 accessions that match those climatic parameters of the accessions we're looking at. And they appear to be, these 10 all in the Andes somewhere. Now, I wanted to do something else. I'm going to say, there's this button here that says show climate. So if I click this button and I click here in Jamaica, I can see the climate for that spot. This again is the same display we saw earlier about the climatic parameters, including the list accessions button. So let's hit that one. And now we get 500 accessions for the with climate are similar to what we clicked there in Jamaica. If we put those on the map, what you'll see, obviously, you'll get some records, some accessions from where we originally clicked, but you're starting to hit accessions in Tanzania and Venezuela and so on, right? So you can use that function, actually even Indonesia. So you can use that function to click anywhere on the map. Let's say something Mediterranean. So let's say I like Corchula, so I'm gonna click here. Oh, sorry. It sometimes does not show the value. And the climatic parameters are displayed. List accessions will try to find things with those parameters plus minus a sensible range. And you can, so this is the full list, but you can adjust your climatic search by climatic parameters to your liking. So Genesys just applies some sensible defaults. So let's say if you say, fine, I don't like 14 and I want 12 to 20. Oh, I'm sorry. I have clicked the lock screen button. So if you want to say 12 to 20 instead of the defaults provided by Genesys, and you hit apply filters, you're suddenly going to be seeing many more accessions because your climatic parameter search is wider. Sorry, I went off too deep into climatic stuff right now.
All right, so for a few accessions, there is no passport data available. How can we retrieve it? And is there a mechanism or tool to obtain this data?
Right, so if you obtain that material from a different gene bank, the best way, or from a different institute, the most convenient way is to go back to them and say, please provide us the passport data of those accessions. If this material just exists and it was there when you joined the gene bank and there's no record of why it's there, there's not much you can do in terms of passport data, but what you can do is properly classify it taxonomically. You can do characterization and evaluation, which is a completely different topic and a topic for a different webinar. So you can do a lot, but you cannot really make assumptions about it unless you obtained it from somebody. Then you can go back to them for the passport data, or use Genesys to try to figure out if the data already is accessible.
All right. So we have so many more questions. I'm not sure we'll be able to cover them all, but we'll try our best. So
let's try.
So is it possible to use Genesys for PGR data information management within a research institute, meaning the information will not be available online for data management purposes only?
No, Genesys is only a database for accession level data that is intended for open and free access. It is not a solution. It's not intended to be used as a tool to manage collections. For that, you have GGC, you have Green Global, you have many different solutions available to deal with that type of need. Genesys is definitely not one of them. It does not deal with the level of detail you need to manage a collection properly.
We also have here a question about the MCPD data. if we can have another run, another go at how to extract MCPD file for the entire collection of a given gene bank, and for each crop and species, and another question similar but by country.
Okay, fine. So let's go views and take who do I remember? Let's take ITC, so Bell 081. Right. So whenever okay, fine. Let's look at let's do it this way. Provenance of PGRFA. So this is a list of countries and international institutes. So let's pick I don't know. I'm not going to click Brazil because I'm going to hit Embrapa, and that's a huge data set. So let's take something smaller. Let's take Bhutan. So what we know here, what Genesys knows is that we have 1,000 accessions that classify Bhutan as the provenance of the material. And those are, well, primarily rice, wheat, some barley, and so on. We know that there are some gene banks in Bhutan, but we have no data in Genesys from those countries. So if you go, again, if you click the number of accession, the number there, it brings you to the known accession browser. It displays active accessions with provenance Bhutan, and from here you can download the MCPD for those accessions. If, on the other hand, you're looking for all data that is published by Embrapa, you would wanna, it makes clear the filters again. You'd wanna filter not by all the different institutes, institute codes. You can say, fine, let's take the records where the country of the gene bank is Brazil. And in this case, that means basically data from Embrapa because we don't have other partners. So you could, in this case, focus on specific crops or specific species. Let's say we want to take soybean and go for accessions and download the MCPD of that crop direct from Embrapa from here. I mean, that is an option. Or if you drop the filter, you would get passport data of accessions in Brazil, and you can still download MCPD even if it's 252,000 records. Similarly for crops, as you adjust your filter, so let's say barley, drop Brazil, apply 220,000 accessions flagged as barley, get your MCPD, do your stuff.
All right, thank you. We have a question about, is it possible to add missing information on accessions in Genesys? And if yes, how? Missing information, missing data on accessions, missing passport data.
Well, I mean, if Genesys does not have Genesys does not have information, then the Genebank does, then the Genebank needs to provide that data to Genesys. So that would help. It does happen that Genebanks share a specific selection of the MCPD descriptors of passport data with Genesys, and maybe not share some details. So it does commonly happen that they wouldn't share the exact coordinates or the country of provenance of their accessions. That does not mean that data does not exist. It just means it does not exist in Genesys because it's not being shared. So assuming the level of documentation in a gene bank just based on what's published in Genesys also is not a good thing to do, because gene banks decide on their own what level of detail they provide to the public through Genesys.
All right, thank you. So we have a question about situation. So how does Genesys handle a situation where two different organizations use the same prefix as institutional code and the naming of accessions?
Right, so the Genesys uses, there's two things. The first thing the Genesys uses is the DOI. So if the accession has a DOI, so a digital object identifier assigned, then that DOI is the unique and primary key identifying an accession. If DOIs are not assigned, then Genesys does one of two things. It uses the institute code of the gene bank plus the accession number, and in some cases, genus, to uniquely identify a record. So if you use, let's say, accession 100, accession number 100 for wheat, and you also have an accession number 100 for barley, then Genesys will needs to use the generic name of that accession to distinguish between the two. And that's a setting on an institute level. So if we would check that out, there's a setting whether an accession is unique within the institute or the same accession number may be used for different genera in the database. So institute code plus accession number and occasionally the generic name.
All right, thank you. We have a question circling back to the mapping. So when you made the selection of lentil and wheat above 2,000 meters, a user could not tell if an attendant could not tell if the species were separated with colors or if all accessions were the same color. In that case, are you able to color the wheat accessions one color and the lentil accessions another without doing individual searches for each species and overlaying the map?
No. So that type of functionality is something you do in GIS software. We can only do the neat and quick things in Genesys, and coloring individual dots is not something we can do, especially because it's very common that you'll have in the same location. So let's do that. You will have in the same location accessions, multiple accessions. So then what color do we give those things becomes a bit of a question. So no, that is not possible directly in Genesys, but you can do that in GIS software very easily. So the reason being, Genesys does not use, so when you filter for accessions, you get 16,000 matches, right? And it does take a while to load those records. And we don't use the same markers that you saw before here, because it would be impossible for the browser to put 16,000 little dots on the map. I mean, it would kill your machine. So what we do is actually we generate, I cannot do it here, but we generate tiles that contain green dots for all those accessions. So if you want to do that with Genesys, you do have to do 2 searches and save one, right? And then display them together, because we cannot color, for performance reasons, the accessions once we generate these tiles. So unfortunately, that directly is not doable at the moment.
All right, we have a question about agroforestry trees and the recording of the passport data in Genesys. Is this possibility? Does Genesys record data for agroforestry trees?
Yes, and the collection of ICRAF is part of Genesys. So just go find it. So World Agroforestry Center, Genebank, number of data sets. So Whitewater's World Agroforestry Center, have a little bit of overview. Fruit trees, multi-purpose trees, different species. Let's look at the accessions. So yes, we do, and Genesys does have tree species in the database.
All right, we have another question about, is it possible to subset germplasm following the focused germplasm identification strategy?
So figs, again, figs is something you can do directly, well, you could do with fig software. We do have an ongoing activity with our friends at in Palmyra, so of the Alliance of Seattle Biodiversity and ICARDA. They are currently working on a figs-like tool that will come to Genesys in the near future. So yeah, that kind of functionality will come in. But actually, going back to figs, if you did, I mean, if you were able to add soil data and other things to the analysis, you would basically come to a similar thing, because what we do here, when we just use the climatic parameters, that already limits the search quite well. So this allows you to do kind of a mini figs, but it's really based only on on climate parameters because we do not have global soil data sets that we could attach to this. But yeah, a subsetting tool by friends in Palmyra and Rabat is coming to Genesys soon, and that will cover more FigC stuff.
All right, you only have a few more questions left. So another one is, what is the objective of having 3 files, collection, core, geography, and name files, instead of one single file in the download of the MCPD?
You're right. So that was not download the MCPD, that was download zip. So that is a Darwin core archive format, which is used by some systems out there. It does require understanding the structure. It's basically kind of a star schema. There's a core file that lists the let's say the main records, and then you have, because you have multiple accessions, or sorry, multiple names for one accession, those names are aggregated in a separate file, and they have a link back to the core file. So you have to do your own kind of one-to-many relationship analysis. So in a way, it is convenient. You have to do a bit more work with it. But in MCPD download, you will get an Excel file and you will not get multiple. You get a single Excel file with the data that you are looking for. So you should expect a file named like this. And this code here is actually the code. So if we look at this filter here, if I download MCPD of these accessions, what you'll see here is that this file name here is an Excel file, and it actually corresponds to the filter code that I'm using here. Why this filter code is, if I take this and I go to another place, this filter code actually includes all my filters that I had applied, right? So you can share those things. So you did not access the MCPD in Excel, you downloaded Darwin Core archive data.
All right, thank you. So is the gap analysis for particular species available in Genesys, and same for collection mission reports?
No, usually gap analysis is a big exercise. Those get published in journals. It's not something we do. We don't maintain that in Genesys directly. We do, however, link to them if we know about them and if we're based on Genesys data. So if you come across publications of relevance, we can promote them or highlight them in our blog posts. What was the other part that was not the gap analysis?
Collection mission reports.
Collecting mission reports. So it is possible to attach the PDF or the collecting data to individual accessions, but there is a big disconnect between collecting missions and samples collected by collectors and what actually hits the Genebank. So we focus on things that are in conservation or are in Genebank in ex situ collections for maintenance, hopefully long-term maintenance. So that you can expect to be able to access that material, or that the material would be available. Now, material collected in the field, once it hits the Genebanks, it still needs to be processed, cleaned, increased, and not all of that stuff makes it. So we do not, Genesys does not currently focus on collecting mission reports. We focus on things that actually has made it through the door and into the Genebank.
All right, so we have 2 questions that circle back on questions you answered. One is about the wheat, especially. There are many genetic stocks developed, for example, monostomics, amphiploid. Will Genesys ever consider cataloging these types of accessions that do not have typical collection data of wild or cultivated vines?
Right. So such material, if part of a Genebank or if accessible through a Genebank, is something that you can expect to find in Genesys. It's not that you cannot. So if we go back to explore all accessions, there is a filter options where you can say, where you can pick the type of the material, right? So forget origin. So you can pick, let's say, a clonal selection or a genetic stock, right? And see what we have about those. So there is a number of such material in gene banks. They do exist in collections. They are accessible. They may not be there for the long term, but as long as the material is in a gene bank and accessible through a gene bank, it can be found in Genesys. And a topic for a different webinar is actually how that kind of information is usually published in Genesys, because it would be part of either a subset of accessions, like the RISE 3K project that was done by Erie. Those you would find under, a list of accessions in the subsets, or maybe even trade data, a section that we didn't even touch in this presentation today. It is something we need to cover in the next webinars.
All right, so we have the other question circling back to the accession names and codes. An example is a particular national gene bank if they use ABC as institutional code in accession naming. And another national gene bank also uses ABC as institutional code. Does Genesys accept such a scenario? Does it not lead to confusion or mix-up?
Right, so no, I mean, it is just looking at passport data, you see which gene bank is maintaining that accession. So accession numbers, ABC1, ABC1, it does happen that those things exist. So let's see, let's just do this. We're looking for accessions with accession number 100, right? There's a bunch of them. So this is the same question, basically. There's no prefix whatsoever because you don't need a prefix. And the accession number just has to uniquely identify the material so that the holding gene bank, know what they're talking about. So if I go to, let's say, if I go to SIAT and ask for their accession number 100, they know exactly which accession to give, right? If I go to Q and I ask for 100, they know exactly what I'm talking about. So there is no conflict of using the same accession number, right? So ABC1. But to properly distinguish between those, especially when there's publications about those accessions, that is why we really encourage gene banks to adopt DOIs, because DOIs make it really possible to globally uniquely reference the specific accession in a specific gene bank and avoid any such problems that somebody would talk about, accession in one gene bank, but people would think it's in a different one. So DOIs and assigning DOIs to material in gene banks is something we encourage. It's not mandatory, but it does really help when people refer to germplasm in collections to refer to the right accessions properly. Because it will happen that you will decide, that the gene bank will decide, oh, we don't like our ABC prefix, we're going to call them XYZ today. So they go and renumber their accession numbers. So with DOI in place, you can label your accessions the way you want, organize them, improve your naming scheme, because the DOI is the key identifier that properly links to the material.
And since we're talking about DOI, the last question sort of relates to this. So what's the status of Genesys? How does it integrate with other international accession plant repositories, for example, FAO-GLIS, FAO-Views, also Erisco and Green Global?
Right, so we do integrate Erisco data with Genesys. The process is not fully automated today. But I am talking to IPK, who maintain Erisco, to help make that better. Green Global does have a wizard available to upload to Genesys. Alelo has so that's in Brapa's system. So Plant Alelo does have an integrated way of pushing data to Genesys directly. So there's no conversion to Excel and BAT. They basically just call the Genesys API and push data in. The integration with the Erie system is similar. They also have their implementation that does that automatically. On the other hand, Genesys does talk to the to the GLIS DOI portal maintained by the Treaty Secretariat. So anytime an accession with a DOI is updated or uploaded to Genesys, Genesys will inform the GLIS DOI service, the DOI registration service, that this accession now is in Genesys, so they can update that registry, and then they do come back to Genesys for passport data and related data to accession, and populate their database to make the GLIS DOI registration service portal more informative and up to date with where people can find, additional information about that accession with that specific DOI.
Okay, no more questions, but Mattia, I didn't relay to you all the thanks that everyone has been sharing in the Q&A, the chat. Thank you so much for giving the presentation today, and thank you everyone for tuning in and for sending in your questions. Just a few notes about future webinars. So today we were only able to cover really a fraction of what Genesys can do and the features. So we will have in the future webinars about specifically subsets, core collections, reference sets, purpose-built sets, trade data, diversity trees that are also now hosted in Genesys, certain tools of validation for passport data, and more on the data management side, how to upload and update data in Genesys. So we will be sharing those invitations with you, and we hope that you enjoyed our webinar today. If you have any more questions, please feel free to send them to the email in the chat, the help desk, also that you see it here. Thank you so much for attending. And with that, goodbye.
Bye all.